No.
All of you. Just to know that the lecture is actually recorded. Yeah,
No problem.
so you will be visible on the recorded lecture at the end. If it
Okay.
doesn't bother you, it's just to let you know. Okay, great. So hello everyone. Back to our lecture. Last time we had a special lecture, a guest lecture, by one of our local data scientists, and which was Fabian Rost, and Fabian explained us from a hands-on perspective, because that's his job, how you can detect odor in non-equilibrium systems. And the non-equilibrium systems that Fabian is working on are, of course, biological systems. And what he basically showed is how odor in non-equilibrium systems manifests itself in low-dimensional structures in these high-dimensional data sets. And he showed you some methods that will also appear on the website. I didn't get to uploading the slides and the video yet. So he showed you some methods of how to reduce dimensionality or how to extract hidden dimensions in these in high-dimensional data sets. And so today, so I will start by giving you a little bit of more introduction to data science and some of the things that we need for the next lecture. That's in the first part of the lecture. And in the second part of the lecture, I will give you another hands-on experience, well, practical example of from start to finish of how to go through such a data science pipeline. Yeah, to start the beginning of the lecture, we'll go back to our New York City flights data set. Yeah, so there's a little gap because we had to find dates with Fabian last time, so there's a little gap. This lecture is now connected to what I told you 2 lectures ago. And let me just share the slide. I'll give you a brief introduction to data visualization. Just a short one, because the slides have been already on the website since 2 weeks, and maybe some of you have already looked at them. Let me just share the screen. Here we go. Okay, great. Perfect. So I'll give you a quick introduction before we go on hands-on example. So today we'll have a hands-on data science example, and next week we'll have a hands-on field theory combined with data science example. Now that will be next week. So today I'll still need to introduce you to some methods that we will need. And if you can confirm that you can see my slides, can somebody confirm? Is that working?
Yes.
Okay, perfect. Yeah, so I'll just give you a quick introduction to how to visualize data. And of course, you're all in your work, you're all visualizing data all the time. But if your data is high dimensional and very complex in structure, it actually matters what you use for visualization. Yeah, and so this 2 lectures ago, when we talked about this New York City data set, about the flights that were departing from the New York City airports, I showed you all kinds of ways of how to do very efficient computations on these data sets. But all these computations, they didn't really give us any real insight. And the reason was that we were dealing with plain numbers, but we never had anything to look at. And today, in the first part of this lecture, I'll quickly show you a plotting scheme, so to say, that is very powerful in visualizing in general and visualizing high-dimensional data. So before that, just a quick reminder. So before we do anything, we always want to make our data set tidy. So we typically collaborate with experimentalists. They give us we obtain the data in a very messy format. And then our first step is to tidy the data. And that means that we need to bring it in a form where every column is an observable or variable, and every row is an observation or a sample.
Sorry to interrupt you. Did you change your first slide?
Yes, you can't see that?
Yeah, no, we cannot see.
I don't know why this always happens now in Zoom. There was a Zoom update. There's something, maybe I have to share the entire screen. Let's try this. The problem is just that I have like 100 windows on my desktop. If somebody has that happens lately all the time, right? That Zoom is not working properly. Okay, let's share the desktop. Okay, now I'm not having embarrassing stuff on the desktop, but that's not the case. Okay, so you should be seeing my desktop now. Okay, a lot of wind. Okay. Now you know what I've been working on. Okay, so can you see this messy and tidy slide? And when I change the slides now, can you see that?
Yes. Right now it works.
Okay, perfect. So this is so just a reminder that the first step that we always do is to make the data tidy. Now, if you have the data in a tidy format, then we can perform column-wise operations that are in most programming language highly optimized and very efficient to run and to program. I introduced you to this very simple R package that allows you to implement all of these operations in data science. And there are many other packages, of course, and other statements. And typically such an operation consists of 3 steps. So you filter the data that is in this data table package, the first parameter. Then you group the columns by some condition. Now that's the 3rd parameter. And then you perform an operation independently on each group, that is the one in the middle. Yeah, so this is a typical step in such a data science calculation. And I showed you how you then can combine these steps along pipelines to perform then complex operations. Today, I want to show you a way of how to more intuitively interact with the data. And the way we typically do that, you all are familiar with plotting, of course. And the way we typically do that is we have a plot in mind, yeah, and this plot then has a name. There's a scatter plot, a surface plot, a histogram, and then you look for the function in MATLAB or so that gives you this specific kind of plot. Yeah. Another way to do that, and that was introduced by someone called Leland Wilkinson in a book, is that you don't give the plots names, but you construct these plots with a grammar. That means that you have a set of rules that allows you to construct step by step almost any visualization of your data. And once you have that, you don't have to remember long names of different kinds of plots. You just add bit by bit, like in a sentence, you add word or there's word by word to make this sentence more rich in information. But the only thing that you need to know is the grammar itself. And this allows you then to create from the simple grammar very different kinds of visualizations. Yeah, and this idea that you have a grammar of graphics, that a set of rules that allows you to construct visualizations, it is in R implemented in the ggplot package, ggplot2, that's quite famous. And in Python, and that's a little bit newer, I don't know how well it works, it's called the plot9 package, that underlies the same idea. Yeah, and in R, we just load this using the ggplot, the library command, and then we are able to use all of these commands in this package. Now, so the basic idea is that we start with a tidy data table or data frame in R. And we take this, and then we assign different visual characteristics of our plot to different columns of this data table. Yeah, so the first thing we have to do is the first thing we have to do is we have to say what to plot, a point or a line, and that's called the geometry. Point, line, bar, circle, whatever. Now that's the geometry. Then we have this mapping that I mentioned, yeah, where we map different aesthetic properties of our plot to different columns in this table here. For example, we could say that the position on the x coordinate should be what is in column A. The y coordinate should be what is in column B. The size of our dots or whatever should be whatever is whatever is in column D, and the column should be C, and the color should be what is in column D. How these values are then translated to specific colors or to specific size or so is a different question. Now we have the aesthetic properties of our plots, of our dots. So where are they located? How do they look like? And then we just have to define a coordinate system to define where they appear on the screen. Yeah, and if you have these things together, we have the simplest version of a plot on the right-hand side. So the way this works in practice is that you have these little building blocks that you just put together line by line. Yeah, so in R, this looks like that you first have to, of course, do something to create an object. And this is just this ggplot command, where you tell the plot what data to use as the first argument. And the second argument is then how to map different columns of your data, of your table, to different visual properties of your plot. And then you add a geometry, for example, point or line, and you have your first plot already. You can, of course, also add more properties. Yeah, you can add more geometries, or you can add more detailed properties of your plot. For example, if you are not happy with Cartesian coordinates, then you can set your own coordinate system. Now you can have polar coordinates or whatever. You can have subplots, you know, by adding this further rule here. It's called facets. You can change how values in this data table, how they map to different properties. So which color represents which value in your table. You can change themes, for example, or different the way lines and whatever are plotted. And you can, of course, also save your file. And these different building blocks of your plots are connected via these plus signs. Now, you put that just as many as you want of these things, of these aspects after each other. And by this, you construct more and more complex plots. Yeah, and for everything that you see here below, there are some sensible defaults, for example, Cartesian coordinates, that in many cases you don't need to touch. Now let's have a little look at our New York City data set. And this New York City data set, right, here we go. Now we already discussed that 2 weeks ago. And in this New York City data set, we have information about flights departing from New York City airports. For each flight, we have different information. We have, for example, the time when this flight departed. We have the origin airport. We have the number of the plane that was used, the carrier, and so on, and the delay for this specific flight. And also, as we already discussed, we can connect this table that we can have and that you can download from our GitHub, from the website. We can connect these flights then to other sources of information. For example, the weather that was the weather information for a given point in time and a given location. We can also connect that to airport information. We can connect that to information about the planes, yeah? And we can also get information about the airlines if we want. Yeah, and that's how what we do. Yes, we load again, just like last time. We load again all of these different files, using the function fread, and then we merge them together using these merge commands. And when we merge them together, we sometimes have to give, for example, when we merge with the planes, or let me see here. For example, when we merge with the airports, that we merge with the airports, we have to say that here the airport identifier is in the column origin, and here the airport identifier is in the column FAA. Yeah? So we merge all of these things together and we have a huge data set, a data table containing all of this information line by line and in a tidy format. Now we already did that 2 weeks ago. Now let's have a simple look at these plots. Let's have a simple plot. And the first thing we can do is, just like last time, we can calculate the average delay for each month. So we group the data by month. And for each month, we take the average over all departure delays and save that average in the column mean delay. Yeah, so what we then get is that for each month, here in the first column, we get a mean departure delay. That is the second column. And below you can see now the simplest plot you can do. You can tell this ggplot function to take this table, now a very simple table, and map the month to the x-axis and the delay to the y-axis. And then you just add a geometry, which is just a point. And then you get what you see on the right-hand side. And you see that something is happening here in these summer months, and something is happening over Christmas, apparently. Okay. So, okay, there's something in the waiting room, right? Okay, so something is happening over Christmas. Now let's go on. We can, of course, also add different geometries to a plot. Yeah, so these different geometries, so we just use the geometry of a line, of a point. We can also add different geometries. And for the sake of simplicity, what I'm doing here, I'm using the tools that I introduced 2 weeks ago, now to do all of these things in one line. Yeah? So we take the flights data set, calculate for each month the average delay, and send everything with this pipe operator here to ggplot. And in ggplot, we just need to define that this aesthetic mapping, that the x-coordinate is the month and the y-coordinate is the delay. And we save this everything, we just save in an object G on the left-hand side. And now we can take this G and add different things to it. We can add different geometries. On the left here, top left, we have the point as before. We can add a geometry line. Yeah, then we get a line. We can add a bar, that's called column. We can add a bar, or we can add all of them together to the plot. And then we have all of them together. This information of what happens with the data, that's not done in the geometry. That's we have done once in the beginning. And now we can just operate on this object and add different things and change the plot the way we like it. Yeah, so there are also geometries that involve analysis. Yeah, for example, if you have a background in biology, maybe then you know the favorite box plot on the right-hand side that summarizes different properties of the statistical distribution. For example, here on the left-hand side, I take the flights, all this combined information. I take it, use the the carrier as the x-coordinate and the logarithm of the departure delay as the y-coordinate, and I add this box plot where I have automatically the median And this is some interquartile range. And then I always forget what this means. It's probably the range of the data without outliers or so. Now that's basically, in some disciplines, these box plots are used to characterize distributions. Another way to characterize distributions are the violin plots that essentially give you a plot of the probability distribution here, just in a vertical manner. The thicker the violin is, the higher the probability to find a data point there. Yeah, and so of course we can now also play with how these plots look like. Yeah, so for example, now if you look at the red, I'm doing the same operation. Yeah, I'm calculating the average departure delay for each month and each airport and each carrier. But now I'm just for simplicity, I only take the big 3 carriers: United Airlines, Delta, and American Airlines. And now I create this plot again. Yeah, I have this aesthetic mapping. The month should be the X coordinate, the delay, the Y coordinate. And now I have another aesthetic, which is the color. I say the color should be the origin airport, and the line type should correspond to the carrier. Yeah, and now I just add the geometry, the line, and I get this plot that you see on the bottom here. Yeah, you can see that all carriers have a plot problem in the summer months and also over Christmas. Yeah, except for something is going on with American Airlines around March. So I have no idea what this is. Yeah, and no, wait, that's not American Airlines. Yeah, that's Newark. Newark American Airlines has a problem in March. Yeah, now you see it's not a problem. The plot is not perfect yet. And okay, so we can go on. Yeah, we can add other aspect. We can change other aspects of the plot. For example, I here say that the fill should be the airport, and then I use a box plot, and then I get an overview over how the different airports compare to each other for each carrier. Yeah, and what you can see is that JFK is doing well for some of them, but not for all. Yeah, for American Airlines and for United and where is it? Delta. Delta is doing well, but there is no clear trend here, of course. Something that's more interesting is if you plot these delays for the big 3 carriers, yeah, as a function of the hour of the day. Yeah, so here the x-axis, the x coordinate, is the hour, and I turn that into a factor. I turn it from numeric to something that is discrete, just for plotting purposes. The fill color is the origin airport, and I added here a subplot, that's called a facet, by carrier. Now, if you remember the 2 lectures ago, this is a formula. We can then use a formula in R to specify how plots are distributed across different subplots. And you can see that for Delta and United Airlines, you can nicely see how these delays add up during the day. Yeah, and it looks a little bit even that this, let's have a look at the next slide, maybe. Let's have a look at the next slide. So if we can have also more complicated subplots, yeah, so for example, we can have a grid by using a more complicated formula where the y-axis, yeah, the y direction should be the origin, and the x direction in this grid should be the carrier. Yeah, and then we get these plots, and you can nicely see how these delays add up during the day. Now, it seems like a little bit, yeah, it's a speculation, but because we have a logarithm in the y-axis, yeah, and we have linear increase over time in these delays over time during the day, that you have an exponential buildup of delays. That's quite interesting. Yeah. Okay. So we can do all kind of other fancy thing. We can, if you have more than 2 variables, Now, for example, when we calculate the average delay as a function of the month, the hour, and the origin airport, yeah, then we have even more variables that we want to visualize. We can do that, for example, with something that's called a heat map. Yeah, and in this heat map here, the fill, yeah, so the color of these tiles is given by the mean delay, while the month and the hour are plotted on these axes here. And then we add the geometry of a tile to get these heat maps. Yeah, then you can visualize relationships between two variables, namely month and hour. Yeah, and it seems like this build-up of delays is specifically drastic in the summer month, while it's not that evident in other months.
Excuse me, I
Yes.
have a question. Syntax. In this, where you've written facet underscore wrap, the first argument tells us the X argument. And so origin will be plotted on the X scale?
Yes, so this facet wrap is just to say, okay, take one column of the data table, yeah, in this case origin, and group the data according to this column, yeah, origin. And then we make one plot for each of these origin airports and put them next to each other, as many as fit on the screen. Yeah, and if they don't fit on the screen, go to the next line. It's basically just this wrap. What I'm saying here is that you have a one-dimensional, so to say, line of plots compared to this grid. That is this grid here. Where was that? Here. This grid is the same thing. It's basically the same thing. So here we have these two coordinates. Yeah, these two directions. Origin airport on the Y direction, and carrier on the X direction. Yeah, this is this formula notation in R. Yeah, so it's a little bit counterintuitive, but you give it a formula in order to tell R how, or this package, how these plots should be distributed on your screen or in the PDF file that you export. Yeah, and you can, if you want, you can use the reason why a formula are used here is that you could do something here that you have here carrier plus, for example, what are we plotting here? Carrier plus month or so. Yeah, you can have here, if you do something like this, you can have more complicated formula to say that a combination of carrier and month of these two columns should be on the X direction here. Yeah, and on the Y direction you have origin. Now you can construct more complicated grids of plots if you wanted to. It's very often that's not very useful, and you can just think about that. That this is what's on the sorry.
That was my question, because in the next slide, the origin argument, the origins have been plotted on the X scale, whereas in this particular case, the origin airport has been plotted along the Y scale.
Exactly. So now I'm trying to get my mouse cursor back. Ah, yes, here. So, okay, now I can change the slide, hopefully. Okay, here we go. So here, I just left away the first argument.
Oh, okay.
I left the left one. So that was originally the Y direction. And now I only have the X direction left, from left to right. And I use here wrap and not grid because I want this these plots, if I have not 3, but like 50 different groups. I don't want them to be all on the same line because I wouldn't be able to see them on the screen. Wrap means once the screen is full, go to the next line. Yeah, nothing else. Okay. Yeah, it's not a complicated thing here. It's just make each plot for each origin, one plot for each origin.
Excuse me.
Yes.
On this heat map, the color bar, the minimum value is not 0. Does it mean that
Yes.
there were flights that departed earlier than scheduled?
Yes, there are. Yes, exactly. They departed earlier.
Oh, it's not very funny for the passengers.
Yes. But sometimes that happens, and it's also a question how the data is recorded.
Mm-hmm.
Depends on how the data is recorded. Yeah. So especially this specifically affects the early mornings, right, and the very late times. Yeah. So these negative departure delays. I mean that sometimes that can happen. Yeah. So sometimes the question is when that's not the time when the gates close. It's probably the time when the airplane starts or something like this. Yeah, and as you know, if the boarding is so very often it happens that once boarding is completed, the airport sometimes the airplane leaves a little bit earlier than scheduled.
Okay, thanks.
But it's a good thing. It's always good to know how the data was actually collected. Yeah, you think that a delay is well defined, but then you can measure this in different ways. Yeah, that's always a very important aspect. And here are also this data set, there are also missing numbers, a lot of missing numbers. And that's when an airport started somewhere but didn't end up on its arrival location, but on some other airports. Yeah, so that's also possible. Okay, so let's go on. Just so we can also use ggplot to do statistical computations on the fly. And this is particularly useful for computing fancy error bars. That's how I use it. And so what we can do, for example, here on the top is that we have the hour on the x-axis, the departure delay on the y-axis, and then color and fill as the origin airport. And then for each of these combinations, because we take on the left the raw data, now we have many different values. We have many different flights. And we can then take just a summary function, statistical summary function from ggplot, tell this function to calculate the mean, and use the geometry of a line to do the computation for us. Yeah, so for so simple things like the mean calculation, that's pretty good. But the nice thing is that we can also do summary functions that are more complicated. Yeah, for example, in this here, we have a bootstrapping confidence intervals. Yeah, that's basically a fancy way of calculating confidence intervals. And we use a geometry of a ribbon, yeah, to visualize these confidence intervals. Yeah, and if you deal with confidence intervals, yeah, you know that's quite complicated to calculate, and then you somehow have to bring them into your plot. So here you don't have to worry about this. You have the most fancy methods just in one line, yeah, and you get a nice visualization of the uncertainty of your data. Another thing we can do is, so here in this uppercase, our x-axis is discrete because it's an hour from 0 to 24, or 23, or 5 to 23. But sometimes we have real numbered values, yeah, and then we need to tell these functions which values to put together. That means we can bin the data. An example here is the temperature in Fahrenheit on the x-axis, and we cannot calculate for every value of the temperature. We cannot calculate a separate mean, yeah, because temperature is a real valued quantity, yeah, and we need to define a bin, yeah, to summarize values of the temperature. And that we can do automatically also with these start summary bin functions, where we just calculate, we just tell the function to bin the data, and for each of these bins, again, calculate the mean and plot the result with the geometry of a line. And we can do the same fancy error bars and confidence interval calculations as before. And also, you know probably that these kind of processes are quite complicated if you have to do them yourself. And here probably the message is that it's not good if it's too cold or too hot. But then there are correlations, right? So when it's hot here, on the right-hand side, there's also when the holidays take place. Yeah, that's July and June. Yeah, in these previous heat maps, yeah, where we had these delays. Yeah, so it's not quite clear whether it's the temperature that's bad for the engines or something like this, or whether it's just the number of people that go on holiday and block the airport and lead to delays there. Yeah, we can do even more fancy things. Yeah, we can do interpolation. Yeah, on the right and the left-hand side here now, I just had the same plot as before. We had, yeah, we have the month. Wait, okay, here's a little error. The hour on the x-axis, and we do yes, the hour on the x-axis. Yeah, and now we can add like an interpolation line just in one line with this summary function. And if we wanted, we would be able to do like non-linear interpolation, linear interpolation, or anything we want just with an argument. And we get our usual nice error bars for free. Of course, if we can do non-linear fits, we can also do linear fits. So we can fit linear models to check for correlations. And that's what we do on the right-hand side. And on this right-hand side, what is actually quite interesting here is that on the x-axis, we have the month. On the y-axis, we have the number of seats in an airplane. And the color is the origin, airport. Now, what we find here is that there's a almost perfect linear relationship between the month and the number of seats. Yeah, there's a perfect linear relationship, which is positive for Newark and JFK, and negative for LGA, which is I think LaGuardia. Yeah, so no idea where this comes from. Yeah, but apparently you are sitting in December in a smaller plane. There's a higher likelihood to sit in a smaller plane in December if you are departing from LaGuardia, while the planes get larger throughout linearly larger throughout the year. Yeah, for some reason. Yeah, so that's one of these things where you should be suspicious and check what is actually the underlying reasoning for this data. Yeah, so that's something I will show you also later, is that it's very important to check whether when you do statistical computations, whether they actually make sense or not. Yeah, the big data gives you every result that you want if you just look for it. Yeah, just because you have so many dimensions, so many samples, you can find every hypothesis you want in these data sets if you just keep looking for it. Yeah, so we can play around with scales. Yeah, so that means that we can change how our plots look like. For example, in this plot here on the top, that's something we have seen before. That's the box plot. We save this in a variable p. Yeah, and now you see why this weird arrow assignment operator in R, why the R community likes that. You can have that, you can assign in the different direction. Yeah? So I have the plot and I assign the results to a variable p. And that's not something, so it's an asymmetric assignment. And so now we have our plot and we can add different color scales. Yeah? We can add different ways of how our data values map to visual characteristics of the plot. For example, we can add a new scale, it's called for some scale color brewer, then we get different blue tones. We can also add like a manual mapping where we say that we want to have black, gray, and white as the colors for our airports. Now, so we can change visual characteristics, and we can also change, of course, how things are positioned relative to each other. Yeah, and I'll go quickly over this because that's a little bit of a detail. Yeah, so we have, for example, we can create this plot here on the right-hand side, where we for each plot, for each month, origin and carrier calculate the average delay for the 3 carriers. And then we can make a plot where we assign the month to the x-axis, to the y, the delay to the y-axis, the fill color to the origin airport, and the transparency of this color to the carrier. Yeah, and then first we can now plot all of this using a bar plot. And if we have a bar plot, we can decide how to put these bars relative to each other. And I'll just give you 3 examples. We can stack them on top of each other. That's on the right-hand side. Now we can dodge them. That means that we put them next to each other. That's in the middle. And we can use a fill position. That means we always fill them up to 1. That means we look at the fraction that a certain carrier and origin airport contribute to the total delays. And let me just see if there's any. Yeah, you can see here, for example, so that here, for example, a large fraction of the delays in March comes actually from this Newark airport, while other months, for example, in the summer month, a larger fraction of the delays actually comes from the other airports, JFK and LGA. Yeah, we can also do something. We can also change how the data is. We can change the coordinate system. Yeah? So now we always assume that we have Cartesian coordinates, but we can of course have any other coordinate system. So here, for example, we are plotting as the x coordinate the wind direction, as the y coordinate the departure delay, and then we just calculate the average delay again using the summary function for a certain intervals of the wind direction. Yeah, and we can then plot that, for example, in different ways. We can plot that, of course, in Cartesian coordinates. But something that's more instructive is actually, when we talk about directions, is to use polar coordinates. Yeah, and you can see that I do that just with adding just one line, one more rule to the plot. And now I can add more aesthetic mappings. For example, I can separate, as before, these different contributions from the wind direction by airport. And this is what I've done here. I just added one more aesthetic mapping here. I said that these bars should be next to each other and not on top of each other or so. And I have the polar coordinates as before. Yeah, then you get the plot that you have on the right-hand side. And in this plot, what you see is that there is a relation with the wind direction of the departure delays, specifically when the wind comes from, what is that, southwest, a little bit southwest-west, for the 2 airports, LGA and Newark. Yeah, whatever is the reason for that. It's actually for all of them. Yes, it's actually for all of them. It's actually for all of them. But it's specifically strong for LGA and Newark. And if you look at the location of New York, that's where the sea is. That's probably also where a lot of the wind, the strong winds come from this direction. Yeah. Okay. Yeah. This was just playing around with the data, and you get some insights from just visualizing the data. And these insights are, of course, much harder to get if you just look at data tables, yeah, on the console, as we did 2 weeks ago. And what you can also see here, if you create such plots, yeah, so you can make more and more complicated plots, but the complexity of your code never changes. Yeah, it does, or it increases just linearly, because you're adding just one bit by bit to your plot, one layer by layer to your plot, and you can make as complicated plots as you want from this. Yeah, without adding more and more complexity actually to your code, or without requiring more and more specialized functions. And that is the advantage of having such a grammar of graphics, yeah, that allows you to have simple rules, visual rules that allow you to add more and more components to a plot. And then of course, we can make these plots look nice. Yeah, we can add things like axis labels for all of our columns. Yeah, typically you get a data table that is where some experimentalist has used their own notation for things. Doesn't make much sense. Most of the time, you want to have your own names for the axis and for the colors and for the legends, and specifically including units if you want to publish that, and then you can do that easily with this labs command. And there's also a title command if you want. Here you can add a title and annotate your plot as much as you want. And then you can get as complicated as you want. You can download extensions. For example, some nice extensions add new geometries, new coordinate systems to these plots. So here, these plots that are used in anatomy, they add the human body and mouse body coordinate systems. And you can then easily, without having more complexity than what I already showed you, you can have visualize your data that you, for example, imaging data on the mouse or human body or whatever you want to do. Now there look like a ton of different extensions to this. Okay, so this is a very efficient way of visualizing data that relies on a grammar, a set of rules. I showed you an R implementation, but there's also a Python implementation. And the Python implementation is rather new, so it's just, I don't know what quality this is. And what we now want to do is I want to show you how to use these tools that we've seen in the last couple of lectures in a specific data science project. And for this, we'll just go through the code of a real data science project. And this is a project that Fabian actually did while he was in the group. And the starting point of this project is a so-called sequencing experiment. Yeah, so I already showed you this table. And here on the x-axis, on the color, the rows in such an experiment, and on such a, that would be, so to say, a matrix that experimentalists would send you. So here, every row is a different gene, and every column is a different cell. Now we have maybe then 20,000 cells, 30,000 cells. And for each of these cells, we have roughly 10,000 measurements. Yeah, and these measurements correspond to how strongly a certain gene, yeah, that's in the row here, is expressed in this particular cell. So these numbers here correspond to how many products of these genes, these experimental techniques, found in a given cell. Yeah, and the way, and these genes, you might heard, they tell us a lot about what cells are doing and how they're behaving and what kind of cells they are. So they're very important molecular measurements of what's going on inside cells. Yeah, so for example, here, this gene that has this ID here, that's a cryptic name, and row 4 is not expressed, it has a little bit information, yeah, in this particular cell, but not in other cells. What other genes, like this one here, have very high expression values. They have very high counts of products from these genes that were detected in these experiments. So what I have to tell you is that these experiments are extremely messy. Yeah, so especially there is a step where the data is exponentially amplified. Yeah, and that exponentially amplifies errors in these data sets. So it's a big mess. Yeah, and now we have to find some structure in these high-dimensional data set, in these genomics experiments. And to show you how this is working, I'll share another file. I'll share another screen. Where are we? Here. Here we go. Now I'll just give you like a hands-on look and on how this actually works. Yeah, I don't tell you too much about the biological background in this project because it's not yet published. Can you see? You should be able to see the browser, right? And you can see that this here is actually a combination of Python, yeah, in the first block, and R. Yeah, so this here is loading some R packages, and here is loading Python packages, and all of this is a Jupyter notebook. Yeah, so you're combining R and Python to take the best of both worlds. Yeah, and then then there's a lot of data loading going on. Yeah, so I'll just go through it, but of course we don't have to look in detail how the data is loaded. Some biological background information about what different genes are doing, and so on. Yeah, and now we start with the pre-processing of the data. So I told you that this data is messy, and this data has like 80% nonsensical information. In other words, this is dominated by technical noise. Now, technical noise is extremely strong, and it gives rise to very weird results. So the first step we always have to do in this particular example in genomics, but also in other data sets, is we have to look at the data and polish it in a way so that we are actually in principle able to detect information here. So for example, this plot here on the top shows you basically what is the percentage of all information in a cell that goes to certain genes. Yeah, they have these weird names and they're completely irrelevant. But you see that this gene on the top here that has an even weirder name, in some cell comprises 80% of the information. Yeah, and that does not make any biological sense, but because if you have 30,000 genes in a cell, it can't be that basically the cell is completely packed with products from a single gene. Yeah, that cannot happen in real life. And that's why we see that here there are a lot of cells. Yeah, so everything where we have more than maybe 30% here, where we don't have any reasonable information. Yeah, that means that we need to do quality control. Yeah, we need to filter out cells that actually have meaningful information, and we have to keep away cells that don't have meaningful information. Yeah, and what we do is we look at such histograms here. So we took a look at these histograms and we calculate probability densities over all cells, so all columns of this matrix. And on the x-axis here is the total amount of information that we have for a cell. So the total number of molecules that we detected for a single cell. And you can see this follows a distribution. And the first thing that you see, there are two blocks. Now, some cells are worse and some cells are better. So there's already some variance in the data just because the quality of our measurement is different between two groups of cells. But all of these are actually good values. Yeah, they're all good. Yeah, and we just take out some cells here, that is the vertical line, that are below 1 million of these counts. Yeah, these we throw away. Yeah, we can also look at other counts. So this is, for example, how many genes did we detect? And here we also cut off. Basically cells that are low quality in these tails here. We just remove them from the data set. Because we know that if we kept them in the data set in the long term, we would have problems with dimensionality reduction. Yeah? These things dominate in the end things that are based on machine learning, clustering, dimensionality reduction, and so on. We remove them from the data set. And now comes, sorry.
Excuse me, can I have a quick question?
Yes.
So is there any, is a systematic way to set the threshold to filter out?
In this case, it's a matter of experience. Yeah, there's not a systematic way. Normally, you would set the threshold in such a data set. You would set the threshold here in the middle between the two peaks. But because the two peaks are both at reasonable values and they are of the same height, yeah, then so we would lose 50% of the data, and that's a little bit too much. Yeah, but they're all reasonable. But we have to check later that if we find two groups of cells in the data, that these two groups are not just representing these two peaks here in the quality of the measurement. Yeah, so later we now go on with analysis, and if there's something suspicious, we go back to this stage here, and we might have to be more rigorous with this cutoff here. Yeah, so in this case, in this particular case, there's no rigorous way of doing that. It's a matter of how much do you expect, what is a good measurement, and basically, now this is a pretty good example in terms of these counts here. Now, if you're working on genomics, so this is actually zebrafish, so we have less than in other animals in total, and but here we don't have basically. Sometimes you have another peak here at very low values, and then we would then completely cut off. So here the problem more is this one here, this plot. We have a lot of cells that have a high percentage of mitochondrial counts. So that are genes or the DNA that is in the mitochondria, and these genes do not produce, this DNA does not produce many gene products. Yeah, so it's suspicious if we have too much of that in this in these cells, and here we take out roughly, I guess, 20 or 30, 20% of the cells we lose in this step. Yeah, and we can also plot both with respect to each other. For example, we can here on the x-axis have these different values that represent the quality of our data, and then just have these bars, these vertical lines that we had in the histograms, just in this scatter plot here, yeah, and cut and see what are the kinds of cells that we lose here visually. Yeah. Okay. Yeah, so now we get rid of the bad stuff. The other things that are totally crazy. Now the next thing we need to do is to make cells comparable. So cells still have different have different measurement qualities. They're still different based on technical reasons. So for some cells, we have a lot of information. Now we have a lot of these counts that we detected, and in other cells we have less. But we want to make them comparable to each other, and that's why we have to normalize the data. Yeah, and there are fancy ways, in genomics, there are fancy ways of doing this, and you can see we're doing all of that basically. And we have to normalize the data to make them comparable. That's what you always have to do. And what we also do here is, because these data counts, you could see like in the matrix that I showed you, there were very large numbers and very small numbers. Yeah, and what we have, so these counts, they live on an exponential scale. Yeah, so these distributions are very skewed. You have a few cells that have a huge amount, or a few genes that have a huge amount of these counts, of these measurements. Yeah, and that's not something that works very well with dimensionality reduction or clustering methods. So we take the logarithm, we log transform the data, yeah, for any further processing. Yeah, that's also something that you have to do if your data is too spread, yeah, that comes from some exponential process or so, yeah, then you have to log transform it. You want it to be something like normally distributed, something symmetrically and rather compact as a distribution. Yeah. Okay, let's go on. So here we do a variance stabilizing transformation. We do more stuff on the data. And then we can go on. Now we can start to understand the data. The first thing we need to do is to see, does it actually make sense what we do, what we have here? Are we actually looking at biological information and real information? Or are we just looking at technical aspects of the experiments? And as a first step, what we do here is we plot a little PCA. Fabian showed that last week what a PCA is. And these data on the PCA, on the principal component analysis plots, they look like this. And you can see, so here I plot, for example, the total amount of these counts in a cell. You know, that's a technical, that's just measuring the quality of the measurement. And you can see there is some variability. Yeah? So these cells have a little bit more, these cells have a little bit more, less. And some of this technical variability is captured by these experiments, by this principal component analysis. So here we're fine with this. It's not extreme. We don't have disconnected clusters, and so we are fine with this. It's already in a good shape. And also we know that in cells can have these differences in the total number of molecules in a cell for biological reasons. Yeah, and then we can look at these plots here. What is the percentage of variance actually explained by certain principal components? Now that's actually the y-axis is something else. But we can order these principal components based on how much they explain the total data. And what we do, and that this is, so to say, like a very professional way of doing things, is we do all other calculations not on the real data, but on the principal components of the data. Yeah, that's an intermediary step that we do just to get cleaner results in the end. So, so what we do here is we take the first 20, 25 or so principal components that constitute like 99% of the variance of the total plot, and we say the rest is noise. That's a way of getting rid of the noise in the data. And now we go on, yeah, and do dimensionality reduction, yeah, further dimensionality reduction. Let me make this larger. Now, I hope you can see these plots. So this is a UMAP. Yeah, so this is a UMAP that is a nonlinear way of reducing the dimensions that Fabian showed to you last week. And you can see once we do the nonlinear dimensionality reduction, our data looks already much more structured. Yeah. So these cells here are actually from the brain. These are brain cells. And of course, there are different kinds of cells in the brain. And because there are different kinds of cells in the brain, we also expect here a structure to pop up in these low-dimensional representations. Yeah, typically these clusters correspond to different kinds of cells. Yeah, so we don't know, that's just a gray bunch of cells here, of dots in the two-dimensional planes. We don't know what the axes are, and we don't know what these cells are. And now we have to dig a little bit deeper. And we do clustering. So here is clustering. And that's Louvain clustering. That's one of these community-based clustering algorithms. And we did that for several resolutions of the clustering. Yeah, so in clustering, in most of the time, you have to tell the algorithm how many clusters you want. Yeah, and that's kind of a resolution that you give to these algorithms. And by doing this, you and what you see here are different clusterings with different resolutions. Yeah, so here you say, okay, give me, what is that, 15 clusters. Then you get this plot on the bottom left. If you say, okay, give me, what is it, 8 clusters, then you get these clusters on the top left. And you can have even more clusters if you want. Yeah, there are different stages. But we don't know yet what makes sense. Yeah, we don't know how many real clusters there are in the data. But we can take all of them and we can go one step further. How do we know that such a cluster is a real biological cluster? We know that if the cells in a cluster all share some property that is not shared by that is not shown by other cells. Yeah, then we know that this cluster is something real that really is going on in the brain. And the way we do that is, let's go down, that we look at the literature. So now we look at the literature, we look at papers. Yeah, so we say we look at papers, and then in these papers, we see, okay, there are different cells and kinds of cells in the brain, and people have done experiments, genetic experiments, for example, where they found, for example, that stem cells express a certain gene. Yeah, for example, this GLULA gene here, that's expressed by stem cells. And now we plot this UMAP with the color representing how much of the products of this GLULA gene we found in a certain cell. And now that makes a little bit sense. And now here in this corner here on the top left, these are our stem cells. And then we can see, okay, so there are also neurons in the brain and many other things. So let's go on. Yeah, so there's another cell type that expresses this cell. Yeah, that's the more advanced cell type from stem cells. Yeah, that's all now in the next step here. That's expressed in these cells here. And then you can go down and identify all of these clusters step by step and identify what kind of cells you have in the data. Yeah, and you can do that with more genes even. Yeah, so there's a lot of genes, like these genes here that identify neurons, different kinds of neurons. So here we have a little just feature of this plot that is identified by this gene. And if you talk biologists, all of these names are associated with different shapes or different functions of cells. We can also do more fancy stuff and to look at different gene scores and as groups of genes, do statistical computations. Yeah, and once we've done that, we decide, okay, for these set of genes here, we have a unique, we fulfill this condition that each of these clusters here, for these clusters, we fulfill the condition that each of these clusters has a certain biological function or represents some biological function because we found a gene in the literature that corresponds to a certain cell type in the body that is expressed in one of them, but not in the others. Yeah, so that's very important. And then we can give these clusters names here, for example, retinal radial glia cells, oligodendrocyte precursor cells, and so on, and neurons. And then you find, okay, so these orange ones here are the neurons, yeah, and here were the stem cells. And then you can start thinking, okay, these stem cells somehow turn here into these neurons as they mature. They get more mature, and then at the end they turn into these neurons here. And then we have other cell types in the brain, like microglia, and so on, that we also can find here. Now remember, in the UMAP, the distances in the UMAP actually, the UMAP keeps the global topology intact. Now that means that cells that are close in here are actually also very similar on in this high dimensional space. Yeah, so it's actually tempting then to think about these paths here as a trajectory that cells take while they go from stem cells into neurons in the brain. Yeah, so let's go on. Yeah, so there's a lot of consistency checks. Yeah, so you have to check all kinds of genes. Yeah, a lot of them, and discuss a lot of with the people who actually know, who do nothing else in their lives but to look at these cells in the brains, and they know all of these genes and all of the papers in these genes. Yeah, and yeah, so do more fancy stuff. Yeah, and then you can do with different clustering. Now we have identification, and this one I said is the one that we can live with these 8 clusters, while for these higher clusters, we have several clusters representing the same cell type. And we can, in principle, come back later to these higher clusters. And typically, biologists want you to get as many clusters as you can. Yeah. So now we have these clusters, and we can have some measurements of how good these clusters are actually. And there are specific plots for these. For example, these are called dot plots. And what these plots show is on the x-axis are gene names, and on the y-axis are the cluster names. Yeah, and the color represents how much a gene, on average, is present in the cluster, and the size of these plots tells you what is the fraction of cells that have this gene on in this cluster. Yeah, so and if we go now here, what we want to see is that these genes that we have here are only on in one cluster, but not in other clusters. Now, for example, a good thing is this one here, this OC cluster. Yeah, there's only one cluster here. There's only these genes here that we have identified for this cluster. It's called marker genes. Yeah, there are computational tools to detect them. Yeah, you only find that in this cluster, but not in the other cluster. Same for this MG, this microglia. We only find these genes in this one cluster, but not in any other cluster. And while we go for, what's the messy one, OPCs, or we go for these neurons here, then it's more messy and it's not so clearly defined. So if we go back now, for example, now if we go here, if we look at these plots, then these MGs, microglia, they were very clean in this plot that I just showed you. Yeah, and that's also represented in this UMAP. There are different cell type that is not produced, presumably by the same stem cells as the neurons. Also, these OCs, as oligodendrocytes, I think, that's separated from the rest. So there's no overlap here. There's a distinct cell type. While for things like we had an overlap between these mature neurons at the 6th cluster, that's called 6 here. There we found that there is an overlap actually in these markers, that they have the same genes that they express, and probably that means that this cluster 6 is an artifact, that we cannot take too seriously. And on the right-hand side, you can see that in the next step, we took these, we went a little bit broader and took this cluster 6 into the neurons here. So now, of course, I showed you basically a finalized version. Normally, you go back between these steps and the earlier steps again and again, back to the quality control until you do not have any trace of experimental technical parameters in your plots. And then also you go back between these clusterings, your experimental friends who do the experiments, and the literature, until you find something that is really corresponding to to what makes biologically sense. It's not that these techniques give you automatically something that there's like a mathematical criterion that would tell you what makes sense. You have to do that, always have to do that yourself. It's not that you push a button and then everything is works automatically, and that's why these people who do these kind of analysis are very much looked for on the job market. Okay, so let me just see if there's anything other interesting. I think there's you can go on and on forever. You know, you can give the experimental lists. Yeah, so which genes are on and off in which cells, and once the experimentalists have these lists, they can do more experiments. They can create, for example, new animals that lack these genes. And what I want to show you is now something that's on the bottom. Yeah, so that the analysis is very lengthy. Now, there are many aspects, of course, that are relevant for biology. I just want to show you a lot of stuff. Now, that's what the biologists are interested in, right? So all these genes. All this stuff, a lot of stuff, even more stuff. Yeah, even more. Yeah, so a lot of calculations. Yeah, more heat maps to check which genes are on where and so on. Yeah, so that's all consistency checks. And one thing I want to just to show you is something that's called, let me see, do we have, okay, is something that's called trajectory inference. You know, that's so these are cells from a brain, and what cells do is they divide and they produce other cells, and then they get more specialized over time. So these cells, they start as a stem cell, and then they mature until they, at some point, they are a neuron. So what we did here is, what you did here and what you do in many cases is you take, okay, I have a snapshot data. And so this fish or these fish were killed for the measurement. I don't have a time course or anything. I don't, it's a snapshot measurement, just one measurement. But I have different cells that are at different stages of this dynamic process of cell maturation. So what then people do is, can we get the temporal information back? Yeah, and this is then trajectory inference of how these different clusters and cell types relate to each other, and we then can calculate rates of how one cell type, the flux that from one cell type leads to another cell type. Yeah, and in principle, based on this, you can then come up with stochastic models that you can, in principle, also compare them to other experiments or theoretical work. Now, that's I just wanted to show you at the end, and let me just go through here if there's anything else that's worth showing you. Yeah, a lot of stuff. Yeah, then you can compare to humans and other animals. Yeah, you can see where there are similarities. These fish that we were looking at are very interesting because they can regenerate their brain, they can build new neurons. And that's something we cannot do so much. So we want to learn where are the similarities and difference. Why are we not able to do that as humans? Yeah, and then we are already at the end of this very lengthy analysis. Yeah, so this is a typical data science project from start to end. And so you can see here a mixture of R and Python. And the important thing is that you cannot, what we showed you in the last lectures, you cannot just go and take the data and throw a UMAP on it or do some machine learning on it. Yeah? So a large part of the data of this pipeline here is to actually clean up the data and think about what is the part of the data that makes sense and what is the part of the data that does not make sense. Yeah, so for example, think about these New York City flights. Does it make sense to have a negative departure delay? There was a That was a very good question. Yeah, it actually makes sense, and this is, so to say, a data set that is used for teaching a lot. So it's already cleaned up a lot, but typically you expect to have a lot of nonsensical measurements in your data. Yeah, or sometimes you have a departure delay of 10 billion years or so. That would be the correspondence in a real data set. Somebody made a typo somewhere, yeah, and then you have that in your data set, and you have to filter it out again. Yeah, this happens all the time. If you are not looking after this, yeah, so if you're not taking care of this, then all of these nice plots here that I showed you, they won't work. Yeah, so they only work because we clean up the data, we normalize the data, we transform the data in a statistical way to make it nicely behaved. Yeah, there's no huge outliers or so. And then these methods like UMAP and so on work on the data. But you always have to do these pre-processing steps before that to make that work. And then next step is you have these fancy methods, but taken alone, they don't make sense. So you can see here how there's trajectories. Cells move here along in time, move along this line and turn into neurons here. And of course, we have many different neurons in the brain. Yeah, and but to understand that, what's happening here in this data set, to make sense of this, yeah, you have to come up with hypotheses. You have to connect these hypotheses with what is already out there in the literature, and then step by step you can construct an understanding about what are actually here the degrees of freedom that you see in this data set. Yeah? And so this is an example, and it's an iterative process that you improve over time. And so this is an example that's a purely data science project. There's very little physics in it. And next time we show you how all of this connects to something that we actually did in the first part of this lecture, namely field theory, phase transitions, criticality, and so on. Okay, great. So I'll stay online, and in case there are any questions, otherwise, see you next week. Bye.
Thank you. It was very interesting.
Yeah. So when are you going